Recent advances in clinical psychology and neuroscience suggest that trauma, childhood neglect, insecure attachment, substance abuse, and metabolic dysfunction stemming from modern diets can collectively produce autism-like neurobehavioral patterns. Although these conditions do not constitute autism in the strict neurodevelopmental sense, they can manifest overlapping traits: social withdrawal, emotional blunting, sensory sensitivity, and cognitive rigidity, creating significant diagnostic confusion and conceptual overlap between developmental and acquired forms of neurodivergence. Evidence from trauma and attachment research indicates that chronic exposure to stress or emotional deprivation reshapes neural pathways involved in empathy, trust, and self-regulation. Dysregulation of the hypothalamic pituitary adrenal (HPA) axis and suppression of oxytocin signaling can blunt affect and social motivation, giving rise to what some researchers describe as emotional immunity, a state of self-protective detachment that may outwardly resemble autism. Similarly, individuals with complex PTSD or dismissive avoidant attachment may adopt rigid routines, monotonic communication, or sensory avoidance as defensive adaptations rather than innate traits. Substance abuse introduces another layer of complexity. Chronic intoxication, withdrawal, or stimulant-induced psychosis disrupts dopaminergic balance and prefrontal function, producing transient states of emotional flatness, compulsive behavior, and impaired social reciprocity. These behavioral changes, often reversible with recovery, demonstrate that environmental and neurochemical stresses can mimic the presentation of autistic traits. In parallel, emerging research on metabolic and nutritional contributors, including type 3 diabetes, advanced glycation end products, AGEs, and gut-derived endotoxins implicates dietary neuroinflammation as a potential driver of cognitive and emotional inflexibility. Diets rich in ultra-processed foods can impair mitochondrial function, induce oxidative stress, and alter neurotransmitter metabolism, producing symptoms such as fatigue, irritability, and sensory hypersensitivity that overlap with the autism phenotype. Taken together, these findings suggest that a substantial subset of adults and adolescents currently identified as neurodivergent may instead be exhibiting acquired neuroadaptive states, functional responses to trauma, addiction, or metabolic injury. This emerging model of acquired neurodivergence highlights the need for more nuanced differential diagnosis, integrating developmental history with metabolic, nutritional, and psychosocial assessment. Recognizing these pathways could prevent misdiagnosis, reduce stigma, and promote targeted interventions that restore neurocognitive flexibility and emotional connection rather than pathologizing adaptive survival mechanisms. Autism is not caused by trauma, neglect, or poor diet, but the appearance of autism-like traits can emerge from the cumulative stress of modern life. Emotional deprivation blunts empathy. Addiction numbs the self. Processed food inflames the brain. Together, these factors create what might be called a synthetic neurodivergence, mimicry of autism rooted in survival rather than neurodevelopment. Recognizing this distinction invites compassion on all sides. It reframes many difficult adults not as broken or defective, but as individuals whose biology and psychology adapted to chronic stress and toxicity. Future research should explore how trauma therapy, metabolic healing, and relational repair can restore the flexibility and connection that such individuals have lost.